In 600 BC, the Romans were using microorganisms to clean their wastewater. They didn't know it at the time since the microscope wouldn't be invented for another 2 millennia, but what they were doing was an early form of a technology we now know as bioremediation. Bioremediation uses microorganisms to remove, break down, or neutralize contaminants in a soil or water source, and is often used in the modern world to clean waste like oils, heavy metals, and other toxic materials. While we humans find these materials toxic, these microorganisms instead see a source of food. But it isn't quite so simple as throwing some random bacteria in a chemical spill and expecting them to get the job done. Not every microbe can degrade every pollutant, so we need to make sure that we use the right ones for the job. Some microbes make specialized enzymes that they use to break down specific pollutants, so we need to make sure that the microbe we're using makes the right enzymes to take care of the problem at hand. Even more challenging is that not all materials are equally simple to break down. Heavier molecules, molecules containing chlorine, branched molecules, and molecules with ring structures all pose an extra challenge to microbes that want the energy contained within it. So let's assume that our microbe has the right equipment. It still might not be able to degrade the contaminant. Even if a microbe has the right enzyme to break something down, other environmental conditions have to be just right for it to grow and do its job. The amount of oxygen present, pH, temperature, nutrient availability, and even the presence of other microbes can prevent it from doing its job. Making things even more complicated is that we often don't even know of a microbe capable of degrading the contaminant we need to get rid of. A large part of research in microbiology involves taking samples in various new environments to try and find new microbes capable of degrading new things. In fact, scientists expect that we've only identified 1% of the total number of microbial species on Earth. And if a substance doesn't dissolve in water, things get even more challenging. A microbe can't eat something that it can't get to, and so typically the food source has to mix with water that's near the microbe. If a contaminant doesn't dissolve, the microbe might be able to take it up at a boundary layer, but this isn't very efficient. So another option for the microbe is to try to get to smaller droplets that might be dispersed throughout the water. A last resort of the microbe's arsenal is to produce a biosurfactant, a soap-like substance that forces the contaminant to mix with water. However, not all microbes can produce a biosurfactant, and a specific biosurfactant might not work on a specific contaminant. Essentially, you really need the right microbe in order to have success with bioremediation. But despite all of the apparent challenges, nature is great at making the right microbes, and we've successfully used bioremediation in multiple situations. When an oil tanker spilled in Alaska's Prince William Sound in 1989, scientists added fertilizer to encourage the growth of microbes in the sound and successfully stopped the spread of oil to uncontaminated areas. And when the Deepwater Horizon oil spill occurred in 2010, there was a panic about the amount of oil that might wash up on the beach. But that never happened. Instead, scientists later learned that oil naturally leaks out of the ocean floor in roughly half the amount released during the spill. The fact that oil was naturally abundant in the environment meant that the needed microbes are already around, and they were more than happy to break down the extra oil provided by the spill. The beauty of bioremediation is that it can restore a damaged ecosystem. But we still have a few reasons to be cautious with the technology since we don't know the effects of introducing foreign microbes into a new ecosystem. Also, it can't totally solve the problem of pollution since it's more of a long-term measure and can't be used particularly well for areas of immediate concern. But despite all of the bad press they get, perhaps it will be microorganisms that save us from some of the greater pollution problems that we'll face in the future.